Hello everybody, welcome to Rayos125, online academic programs organized by ISRT Kerala chapter. I am Jaseem, Advanced Training in Medical Imaging Technology student, Regional Cancer Center, Trivandrum. Today I am here to present on the topic diffusion weighted imaging. Diffusion weighted imaging is a magnetic resonance imaging method that produces magnetic resonance images weighted by the local characteristics of water diffusion. Diffusion means random movements of the water protons. The process by which water protons diffuse randomly in the space is called as Brownian motion. Movement of water protons generate contrast in magnetic resonance image within a voxel of tissue. This motion is restricted by boundaries such as ligaments, membranes, macromolecules, and diffusion is also restricted in pathology. Types of diffusion: isotropic and anisotropic. Isotropic diffusion: possibility of water protons moving in any one particular direction is equal to the probability that it will move in any other direction. Isotropic means uniformity in all directions. In anisotropic diffusion, diffusion has preferred direction. Water can move easily in one direction than other. Principle of diffusion weighted imaging. Diffusion weighted imaging is a T2 weighted spin echo sequence with the diffusion gradients applied before and after 180-degree pulses. Now, diffusion gradients can be applied to various sequences, mostly applied to echoplanar imaging sequence with the infinite T2. Steps of diffusion weighted imaging. After 90-degree pulse, protons aligned. Then first diffusion gradient applied, variable phase shift along the gradient. If protons do not move, 180-degree pulse and second diffusion gradient applied and complete reversal of phase, it gives bright signal. If protons move, 180-degree pulse and second diffusion gradient applied, not complete reversal of the phase shift by 180-degree pulse and second diffusion gradient gives a reduced signal. Terms and concepts. The B-value. B-value indicates the magnitude of diffusion weighting provided by the diffusion gradients. It also indicates sensitivity of the sequence to the diffusion. B-value expressed in second per millimeter square. B-value depend on amplitude, separation, and duration of diffusion gradient. The B-value increases with the diffusion gradient strength, duration of their application, and time between application of the two gradients. As B-value increases, the signal from the water reduces. Diffusion trace. Isotropic diffusion form the basis for the routine diffusion weighted imaging. Also, there will be some anisotropic movements of water as well as, especially in brain from white matter tracts. To reduce this anisotropy, the image with a higher B-value, like B-value equal to 0, is occurred in all three directions, X, Y, and Z axis. Diffusion changes along all three axes are then averaged to get a trace diffusion image. ADC, apparent diffusion coefficient. The net displacement of molecules diffusing across an area of tissue per second is called apparent diffusion coefficient. ADC calculated mathematically from B-value equal to 0 and a higher B-value image. Signal attenuation of a tissue with the increasing value plotted on graph with the relative signal intensity on y-axis and B-value of x-axis. The resultant slope of line is apparent diffusion coefficient. ADC is done pixel by pixel basis by computer. To user it is available as ADC map. ADC is independent of magnetic field. Reduced ADC is restricted diffusion on diffusion weighted imaging, while on same area will be dark on ADC map. T2 shine through. Signal intensity on diffusion weighted imaging not only depend on ADC, but also on T2 relaxation time of tissue. High T2 appears bright on diffusion weighted image, even not restricted. ADC map help to differentiate T2 shine through from actual restricted diffusion, or T2 shine through are exponential images formed by a ratio of diffusion weighted images divided by T2 weighted image in same series. These images are called EADC by some vendors. Truly restricted area is bright on EADC. Technical parameters of diffusion weighted imaging. In brain imaging, TR in the range of 5000, T80, number of excitation 4, FOV 22, frequency direction right to left, slice thickness 5 mm and B value 1000. Body imaging, TR in the range of 7000, T80, number of excitation 6, FOV 30, frequency direction superior to inferior, slice thickness 4 mm and B value 700. Clinical applications of diffusion weighted imaging. Neuroimaging applications. In stroke, failure of sodium potassium ATP pump tissue ischemia result in influx of extracellular water into cells. This is called cytotoxic edema. Net shift of water molecules from extracellular into restricted intracellular space. Overall reduction in diffusion of water molecule in that area. Manifested as a bright signal on diffusion weighted imaging and a dark signal on ADC map. Diffusion weighted imaging can detect early ischemic tissue. Diffusion weighted imaging show stroke lesion earlier than other sequences. The picture A, diffusion weighted imaging shows bright area in the right parietal region. On ADC map, image the area turns black, suggestive of acute infarct. Epidermoid versus arachnoid cyst. Epidermoid is a composed of keratin, debris, and salt cholesterol, provide hindrance to diffusion of water molecules. Epidermoid is seen as a bright lesion on diffusion weighted imaging. Arachnoid cyst is a clear series of containing cyst. It will not be bright on diffusion weighted imaging, will be same as CSF in the signal density. Diffusion weighted imaging can detect a residual epidermoid. Abscess versus simple cystic lesion. Abscess contains thick fluid with a hindrance to water diffusion. It shows restricted diffusion predominantly in the sphincter. Cystic lesion with a relatively clear fluid does not show restricted diffusion. In figure A, flare may show a large abscess in left cerebral hemisphere. Diffusion weighted imaging show central part of the abscess to be bright. In ADC map, central part of the abscess turned black, suggestive of restricted diffusion in ADC. Diffusion weighted imaging in brain tumors. Diffusion weighted imaging provides qualitative and quantitative information at a cellular level, such as cellularity and cell membrane integrity. Tumor with more number of cells and intact cell membranes not allow motion of water molecules and appear bright on diffusion weighted imaging. Less cells and broken cell membranes not be restricted on diffusion weighted imaging. Diffusion weighted imaging used to detect, characterize, and to assess chemotherapy response in tumors like medulloblastoma, ependymoma, and lymphoma. This is a case of lymphoma. Figure A show left cerebral hemisphere bright on diffusion weighted imaging and dark on ADC, suggesting a restricted diffusion of water molecules due to high cellularity. Diffusion weighted imaging in body. Use of diffusion weighted imaging in body imaging is a relatively new technique. Big obstacle in diffusion weighted imaging in body imaging are motion and short T2 of various organs. Imaging done with breath hold and respiratory triggering. Diffusion weighted imaging mainly used in tumor imaging and in follow-up imaging. For staging tumor and lymphoma, whole body imaging with background suppression is used in replacement to PET imaging. Diffusion weighted whole body imaging with background body signal suppression. Images show only diffusion restricted structures and tissues. Advancement of diffusion weighted imaging. Diffusion tensor imaging, fiber tracking maps or fiber tractography. Routine diffusion tensor imaging based on anisotropic diffusion of water molecules. Tensor is a mathematical formalism used to model anisotropic diffusion. Technique. Magnetic resonance scanners axis X, Y, and Z are now perfectly parallel to the white matter tracks at every point in the image. In diffusion tensor imaging, images are occurred in at least 6, usually 12 to 24 direction instead of 3 in usual trace diffusion. Pure ADC for each pixel is calculated from these images in multiple directions. This is called the principal eigenvalue. Principal eigenvalue is calculated along the true axis of diffusion called eigenvector. Images formed with the principal eigenvalue is called diffusion tensor imaging that gives orientation of fiber tracks. Uses. Diffusion tensor measure the magnitude of the ADC in the preferred direction of water diffusion and also perpendicular to the direction. The resultant image show white matter tracks very well. This technique is called tractography. Various maps used to indicate orientation of the fiber track include fractional anisotropy, regional anisotropy, and volume ratio maps. Tractography for the assessment of relationship of tracks with the tumor invasion of tracks, and preoperative planning. Diffusion tensor imaging used to evaluate white matter tracks in various congenital anomalies and dysplasias. Thank you all for listening to this presentation.